Okay, this is David Zierler, Director of the Caltech Heritage Project. It's Monday, February 7th, 2022. I'm delighted to be back with Dr. David Ho. David, as always, it's good to be with you again.
Good to see you again.
David, what I'd like to do today is, before we go back to those very early days of the HIV pandemic, I wonder if we could superimpose what you were seeing in real time then with all that we have learned about HIV and your contributions to it in the meantime. So, just as a basic primer, when someone is infected with HIV, what are the biological processes that lead to symptoms, illness, and if left untreated, death?
So, if you're asking about what we know today, I could give you that. HIV gets into a person usually via sex, and occasionally via blood transfusions and such. But the virus gets into a particular cell that we call CD4 lymphocytes, and these cells are a very important player in our immune system, orchestrating a number of different critical functions. And what we have learned over the years is that the virus infects that cell and primarily that cell, very few other cells, and just from the outset starts to replicate in those cells. And initially, in the first, say, 2 to 4 weeks of infection, there's a burst of virus that typically results in a flu-like illness. And most people simply attribute it to another viral infection. And then that illness will self-resolve, and the virus is brought under control at varying levels depending on the person. And then that person is asymptomatic and goes on for many years, if untested, not knowing about the infection status. And I said earlier, at varying levels, if the virus remains at higher level, the person will develop AIDS faster. If it's at a lower level, it will develop slower. And what determines that balance is a reflection of the immune response, as well as the characteristics of the particular infecting strain. But typically, it would take somewhere around 10 years for a person to go from infection to development of AIDS. And that's because during this prolonged period, while the person is essentially asymptomatic, the virus is cranking away and slowly depleting this important cell population that is so vital to coordinating many important immune functions. And so that's what we know. And about 99% of the time, if left untreated and if followed long enough, the infected person will die from AIDS. So it's an extremely lethal disease if untreated. But it's also very insidious. It takes many, many years for the disease itself to manifest unless you're doing testing.
And what exactly kills a person when they're left untreated? What is that mechanism?
Typically, it's not HIV itself. Most of the time, once the CD4 cell number drops below a critical level, then that person is prone to develop many other infections. And many of them are infections that normally do not do much harm to a healthy person. But when the immune system is depleted to an extent, then they would develop bizarre infections. Last time we talked about pneumocystis, which is only seen in cancer or transplant patients, and now HIV patients. That would be common. Normal people don't get that kind of infection. So they're what we call opportunistic infections, And you could name a couple dozen different pathogens that would infect the lung, the brain, the gut, the eye, and many other organ systems, simply because they are opportunistic. They only matter when the immune system is down.
Just theoretically, if somebody lived in a hyper sterile environment, the ultimate lockdown with masks and all the rest, could somebody live with HIV a healthy life if they're not, you know, being being exposed to illnesses for their weakened immune system?
Well, unless they lived in the bubble from childhood, from infancy, in fact, because many of the viruses that plague HIV, many of the pathogens that affect HIV patients are ones that are already in the person's body. For example, a cytomegalovirus infection, most people have acquired that during childhood. And when the immune system is down, that comes back. And that could cause retinal destruction, could cause hepatitis, could cause gastrointestinal problems, and could kill. Um, shingles, for example, could appear and it could be disseminated, and that's because of childhood chickenpox. So there are a lot of these things that are already in the system that you can't do anything about, and they're also ubiquitous in the environment. Um, and so yes, indeed, HIV, if you let it continue, could also cause the person to lose weight and also could do a lot of harm. But that typically comes only after the opportunistic infections have already occurred.
Well, David, now let's go back to the early days in Los Angeles. Was your sense then, in retrospect, that if this virus can incubate and a person could be asymptomatic for many years, that the initial patients that were coming to you had been infected not weeks or months before, but even years prior to interacting with you first?
That is absolutely correct. That they were infected many years before. Of course, these could be the ones that developed disease first, right? I mentioned that varying high levels would manifest itself, high levels of virus would manifest itself earlier. So we probably seen people who were infected for 3, 4, 5 years rather than 10 years at that time point because they came in early in the process. So there's always a bell-shaped curve in terms of distribution of when one would develop disease. And so those people were, that they came in late 1980, 1981. They're the earlier cases. And they were undoubtedly infected a number of years before. So probably by the 70s, the virus was already in the country, probably spreading undetected.
Was there initial concern about transmissibility and the way that healthcare practitioners might need to take special precautions? In other words, were people talking about masks or extra layers of protection simply because it was not understood how this transferred from one person to another?
Yes, I would say in the very beginning, the first 6 months or so of recognition of this syndrome, there was a lot of fear, and even fear among healthcare workers and others who are involved in the care of these individuals. Since it was so mysterious, we didn't know very much about it. However, for those who sort of look into the disease more deeply, it was clearly a sexually transmitted disease at that point. Now, by a number of months after the initial detection, there were some cases that were not fitting in very well. In particular, Haitians who were not gay men were coming. I saw one such case in 1981, for example. that didn't fit the bill, and yet had. And so there was some concern that there may be other routes of transmission. Of course, in retrospect, we know we understand the Haitian connection, but at that time we didn't know whether it was the Haitians were acquiring it by sex or not. In terms of and then again, some months later, there were cases of hemophiliacs and blood transfusion recipients, and that says it's sexually transmitted and bloodborne. And so the sort of 6-9 months into the early part of the visible part of the epidemic or pandemic, it was believed by the experts who have been looking at this that it's probably not casually transmitted and more likely blood and sex related.
David, I'm curious, before you and your colleagues reached the conclusion that it was not casually transmitted, but it was sexually transmitted, if you were aware of concerns earlier in the 1970s that led to the creation of, for example, the Asilomar Conference that David Baltimore and others led, stemming from the concern that with the recombinant DNA revolution, all kinds of scary things could happen in a laboratory research environment. I'm curious if you thought about any possible connection in the very early days between this new syndrome and something that might have come out of a lab by accident.
It didn't cross my mind. What crossed my mind was this looks like a new pathogen. And it was that in itself was already enough of a mystery. As to its source, obviously, in the beginning, we had no idea. We just knew that it was being transmitted among gay men, and then the blood recipients or hemophiliacs who received blood products. We knew gay men were a major donor population for blood donations. So that, and obviously we didn't test for that at the time. So that explained why we were seeing the infection in certain individuals. But in terms of the source, that remained mysterious for a long, long time. But it didn't cross my mind that this was engineered or leaked or anything like that from a laboratory.
David, once it became clear that this was primarily a health crisis in the gay community, were you involved at all in campaigns to raise public awareness, outreach with community leaders, that this was something that these people should be concerned about?
I was not that involved in that aspect because, quite frankly, I was a pretty junior scientist, and I had things to do in the lab, things to do in the clinic, so I pretty much focused on what I needed to do. Now, of course, I follow with great interest what was going on in society, and I read about the fear within the gay community, and how mistreated and I could personally witness how mistreated the patients were, right? They were shunned by healthcare workers. They were shunned by families and friends, because, people had a lot of fear about this new disease. And it was clear that these young men were dying very quickly. And so everyone was afraid to catch this. And you asked about masks and so on. Of course, until it became clear that this was a sex and bloodborne disease, all the precautions were taken. The healthcare workers were gowning up, wearing masks. And I should say, many of the patients actually had TB, which is also an opportunistic infection. And so masking was mostly appropriate for that. But in retrospect, inappropriate for HIV itself.
What therapies were available? What were you thinking of minimally to make the patients as comfortable as possible, if not even to search for a cure? How could you prolong their lives to the greatest extent possible at that time?
Well, fortunately, many of the what I call opportunistic infections were treatable. Last time we talked about the use of a drug called pentamidine to treat pneumocystis pneumonia, and there are other drugs to treat that pneumonia as well. So we could take care of the acute problem that resulted from the immunodeficiency, but we could not treat the underlying disease process. And I think all of us knew that. And all of us knew that we can't treat that until we find the cause, right? And the cause was essentially unknown until 1983, 84, that period when a retrovirus was first identified to be linked to this disease in 83. And by 84, it was pretty certain that retrovirus was the actual cause.
Now, in the senior year in your residency, you mentioned last time you were in the lab at UCLA. You were working on unraveling this mystery. So let's talk a little bit about that. What's the biggest mystery? What is the fundamental thing you're trying to figure out? And then how do the questions get more specific and targeted from there?
Let me correct the record a little bit. Yes, in my senior year and senior residency year and chief residency year, I was at UCLA, but I was doing mostly clinical work.
Okay.
I did not get to the lab until 1982 in Boston.
Okay.
And that's where I began to focus in the lab on this problem.
All right, so now let's go to the decision making. You returned to Boston in 1982. Where are you going first?
So I went straight into an infectious disease fellowship at Massachusetts General Hospital and Harvard Medical School. And I worked in the laboratory of Martin Hirsch. And Marty is a virologist. He worked on cytomegalovirus and herpes simplex virus and others. Obviously, this arrangement was made about a year prior to my going. So, AIDS was already on the radar, and he, like me, was quite interested in this new disease. And because of that mutual interest, I was the first one he put on the project to study these patients.
Do you have a clear memory of when the terms HIV and AIDS came into use?
No, AIDS it went by many other names, but a sort of a nomenclature committee later, about a year into the disease recognition, it was AIDS was named. Prior to that, it had many other names, and some are rather derogatory. But GRIT was a common one, that gay-related immunodeficiency. Of course, that turned out to be largely incorrect as the pandemic exploded throughout the world, that it's certainly not largely gay-related, except in the US and Western European countries.
Now, to give a sense, you said that by this point, when you got back to Boston, this illness, this syndrome was already on the radar. But how much was it really? In other words, were there many places you could have gone to pursue this interest? Or was Hirsch and that lab really pioneers in this field?
No, there were pioneers in virology, but in a different discipline. But at that point, we didn't know what virus or what agent was causing this, right? And of course, there are many laboratories throughout the world doing virology. Some working on herpes viruses, other working on influenza viruses, and other working on retroviruses that up to that point largely did not were not associated with human disease. And so when you don't know what category the pathogen fits into, you cannot pick a proper lab. Uh, so you go to a good virology lab, and hopefully that will, uh, uh, be the base and the foundation for the work.
How did you first connect with Hirsch? Were you just generally an admirer of his work?
Many reasons. One is, I wanted to do my infectious disease training in a good place, and the program at Massachusetts General Hospital is one of the top in the country. So that form a big reason for wanting to go to that institution. And within that institution, Marty was the leading virologist. And during the interview process, I talked to him, his colleagues, and others in the lab, and I felt that was a great place for me. Similarly, I almost went to a program at the Fred Hutchinson Cancer Research Center in Seattle that also had a strong program in infectious diseases. And a number of people were involved in virology there.
Now, did Hirsch have direct contact, or did he interface at all with AIDS patients himself?
Yes. Yeah, he's a physician scientist, so he has both laboratory and clinical background.
So when you get there, what exactly is the project, and who's funding it? I think that's important to see who's putting effort into supporting this research in this early period.
Well, Hirsch did not have funding for AIDS because it's new. Very few people had. So he had funding for his virology. So it's sort of like robbing Peter to pay Paul here. So basically, we just used whatever resources he had at his disposal, and we started to look at patients, and in particular, one aspect of the AIDS complication was a problem which we call Kaposi sarcoma. Kaposi sarcoma has been long recognized in Africa as a skin cancer with dark spots all over, and sometimes in the mouth and the gastrointestinal tract for severe cases. And in the first few cases of AIDS, we already knew some of them had Kaposi sarcoma also. And there was always an impression that Kaposi sarcoma was transmissible and therefore had a viral cause, perhaps. And in fact, there was quite a bit of literatures to suggest that it may be a virus and it may be in the herpes family. And so since Hirsch lab was focused on herpes family of viruses, the first problem I focused on was Kaposi sarcoma that was being seen in the AIDS patients, to see if there's a cause for the Kaposi sarcoma seen with AIDS, because that would link the AIDS problem to the expertise of the lab. And years later, this is not my discovery, years later, scientists at Columbia actually found that it's due to a herpes virus, which is, was termed herpes 8. That's what the 8th herpes virus discovered in the field. So that suspicion turned out to be true. But because I then was working on samples coming from these patients, I took a great interest in what was causing the underlying condition rather than what was causing Kaposi sarcoma. And so I started to take samples to look for a virus. And of course, we didn't make that discovery, right? The scientists at Pasteur and scientists at NIH in 1983 and 84 really identified the virus and went on to show that definitely this new retrovirus was the cause. But my work was behind. And so those two groups were first, and another group at UC San Francisco also within a month or two reported the isolation of the virus, and our group was the fourth. So we were beaten, but it taught me a very important lesson that you could be working hard and making the same discovery, but even if you're a week late, your work would be undervalued a great deal because it's already known to other people through other endeavors.
Did you feel like you were in a race? Was it a friendly competition? Were you aware of what was happening in these other research groups?
Oh, yes. I mean, a lot of virology labs throughout the world were looking at this, so we knew. But, if you look back, the people who discovered this virus were retrovirology labs. That makes sense. And so it was the retrovirology lab at the Pasteur Institute, the retrovirology lab at the NIH. And so, they had all the methodologies already, and no surprise, the UCSF lab was a retrovirology lab. So we were the only non-retrovirology lab to quickly, and we caught up very quickly. I mean, our publication was only a couple months later. Of course, by then, people already knew what was the cause, and we sort of just added to the record.
Now, the samples you were studying, what were they samples of, and what were you seeing?
We were studying blood samples, of course. We know the virus is in the blood based on the clinical information. We were studying genital fluid, semen, and other genital secretions. We know the virus has to be there based on the clinical information. And we were putting it on cells to see if something will grow out that we could identify as a new agent. And we also know that the virus seems to cause a destruction of these CD4 T cells. So the natural thing to do was to try to grow an agent using CD4 T cells that we derive from blood donors, normal donors. And that's exactly what the Pasteur scientists did, NIH scientists, and the UCSF, and we were doing the same thing, except we didn't have all the tools that they already had in the lab. For example, David Baltimore's Nobel-winning discovery of a reverse transcriptase. Those assays were up and running in retrovirology labs, but not in our herpes lab. And so I had to sort of reinvent that in order to do what the other labs did. And the other labs had, the NIH lab had maybe 50 to 100 people. The Pasteur lab had probably a group of 20 people. And I was doing this by myself with one technician. So there was no competition. They of course were way ahead. But once the virus is discovered, I did a lot of things that that a physician scientist would do, right? So I could quickly show that, for example, that the illness that I talked about at the very beginning of infection, that was due to HIV. And I think the work I did was the first to show that HIV acute infection could cause a transient illness. And that's the first sign. and then the people become asymptomatic. And then the work I did, published in the same year as the NIH papers in 1984, showed that the virus is carried by asymptomatic people. So there's a carrier state. So there's a carrier state, and that's, of course, very important for transmission, right? The person is carrying the virus and could pass the virus on, even though he was not sick. And at the same time, we show that the virus is present in semen. And that makes all the sense because of how this disease is transmitted. And then because I was not only doing the lab work, but I was seeing these cases in the clinic and in the hospital. There were a lot of neurological problems seen in the AIDS patient, and some of them are due to the opportunistic infections. But even absent that, there was a lot of neurological deterioration, and it was clear that there was more than just immunodeficiency. And so I think our group, again, was the first to show that these neurological problems were a consequence of HIV infection. Mm-hmm. And we were, from the postmortem cases, we could find the virus in the brain, in the spinal cord, in the spinal fluid, and show that late in disease, the virus would go on to destroy the neurological system as well. And that's because the virus infects another second cell type called macrophages. And macrophages could move into the brain and turn into what's called microglial cells, and somehow indirectly cause the damage to neural cells. And how that occurs is still unknown today, believe it or not, after all this time.
David, in that time frame, 1983, 1984, how well developed is the subfield of retrovirology?
Retrovirology was of great interest to many basic virologists who were trying to understand what retroviruses do in animals. And in fact, retrovirology was of great interest because many of these animal infections due to retroviruses result in cancer. And no doubt, that's why David Baltimore was working on retroviruses and reverse transcription. And so there are many retroviruses that cause leukemia, lymphoma, and other cancers in various animal species, but not humans. But by the time AIDS appeared, almost concurrently, it was being discovered that, and this came out of the NIH, that there is a very rare lymphoma cancer in humans that was occurring in places like Japan and the Caribbean. And so Japanese scientists and the NIH team that I spoke about under Bob Gallo discover a human retrovirus called HTLV1 that was associated with that human lymphoma, which was called adult T-cell lymphoma. Interestingly, it also infected T cells and caused T-cell lymphoma. And so I think that was the beginning of human retrovirology. Prior to that, there were no retroviruses causing human conditions.
Now, you mentioned earlier that your competitors at Pasteur, UCSF, and elsewhere, there was an unfair advantage methodologically. So let's start first with the instrumentation. What did those labs have that you were not working with?
I think most importantly, a routine assay for retroviruses was to see if something is spreading in your tissue culture, and then detect the presence of reverse transcriptase activity. So that was the key. And that's a biochemical assay that Baltimore and Temin developed in the 1970s, right? So that's a routine assay in retrovirology lab, but not one in a herpes laboratory. And so I had to spend quite a bit of time. And I also did not have a lot of biochemistry background. I mean, I knew my knowledge in biochemistry, but in terms of hands-on experience, I wasn't all that savvy with such assays. So it took a while to get that going, and that was the key. And also the fact that, you know, the other teams have lots of people. And I was a junior postdoctoral fellow, basically, working on this. Whereas the rest of the lab was working on herpes viruses. And so what would have been really useful was the reverse transcriptase assay and electron microscopy, you know, that would be readily available in a place like the Gallo lab or the Pasteur lab.
What about intellectually, David? What kinds of questions were people in retrovirus labs thinking about that might not have even occurred to you at that point?
Yeah, I think probably not that much more. I think it's mostly methodology and familiarity with retroviruses and how the genes are organized. Obviously, they had more in-depth knowledge about all those things, but I think it was mostly the availability of the tools. You know, we all read the literature, and by then we knew that there was one human retrovirus, and that happened to infect T cells. And coming back to the nomenclature question you asked earlier, this is why initially HIV was called HTLV3 by the NIH group, and that name stuck for a number of years, even though HTLV3 or HIV today is not that closely related to HTLV1 and 2. HTLV1 caused the adult T cell leukemia that I mentioned, and some years later, HTLV2 was discovered, and that caused another kind of, we call it hairy T cell leukemia. And so even those of us who did not work on retrovirology could make the connection, hey, there's already a human retrovirus that infects T cells. So this disease, if it's a retrovirus, we should look at T cells. And so that didn't take much to connect the dots.
What did the actual discovery look like? What was it, a eureka moment? What was identified that made this clear what this syndrome was?
So I'll tell you what the Pasteur group first reported. They first reported in a few cases the detection of the reverse transcriptase activity in their cultures. And then they show a virus particle on their electron microscopy. And that's pretty much it. And it was not convincing. And in fact, if you look back at the electron micrograph from that publication, in retrospect, it doesn't even look like HIV. But they are the first to detect the reverse transcriptase activity. And they reported that. And that's why the Nobel Prize in the AIDS discovery was awarded to the Pasteur people, and not to Bob Gallo at NIH, and not to others that came later. And what the Gallo lab did was to detect the retrovirus in convincing ways, in ways that the Pasteur group had not done, to grow the virus and to show the reverse transcriptase in robust fashion, and that is passed on, and they isolated not just one, but many such viruses from dozens of cases. And then using the virus that's then isolated to develop a blood test to show people who had AIDS almost always have antibodies to the virus. And those who are not, and not just healthy carriers, but the healthy individual, normal donors don't have antibodies to this virus. And they went on to show in very convincing way. That's why I said the discovery was really, there was not a eureka moment in the sense that the Pasteur group provided a hint, which was fully developed by the Gallo group. And I'm sure that involved an army, and they published 4 papers simultaneously in Science. And in fact, you could tell about 100 people worked on that.
Now, the people who published before you, given that your paper came out 4 months later, were you influenced by their research? Were you confirming what they saw? Or were you able to draw new conclusions based on what your colleagues elsewhere were finding?
I think I was mostly confirming. I think certainly by the time that the Pasteur group was, like I said, not convincing, but provided a hint, a clue, and certainly motivated me and others to look even harder at retroviruses. By the time the Gallo NIH papers came out, we pretty much had something on a few cases that would confirm their results, but independently generated. But we were clearly months behind their work. And so they convinced the world already. And so for us, yes, we published what we had, but we also knew there were plenty of other unanswered questions that we could address. And, this is, so once the retrovirus was clearly established as the cause, there was a lot of low-hanging fruits to be picked, right? Especially for someone junior like me, who also is bridging the clinic and the bench. So that's why I told you all those things earlier about the discoveries that we made in the 1984 period, about the acute infection, about neural invasion by the virus, about, curious state of asymptomatic individuals. All those we did in short order, and really propel our lab into the sort of the active echelon of folks working on human retroviruses.
David, before we proceed to the next steps, what's able to be considered now at this point, let's zoom out to the national level. What's happening in the Reagan administration, at the NIH, at the FDA? What's happening that suggests that there is at least the beginnings of a national response to this crisis?
Well, I think I would give a fair amount of credit to CDC, which recently has been criticized for a number of things in COVID. But in retrospect, I think CDC did a great job of tracking this new disease, making everyone aware of it, and keeping a head count. And they brought a lot of attention to it. And some of the leaders at CDC were leaders in the field. The Reagan administration, on the other hand, was largely silent. This was a gay disease. You know the conservative nature of that administration. And you could say it wasn't until the end of the Reagan era did the administration utter the words HIV/AIDS. So yes, NIH responded and Congress to some extent, but it did not received the full blessing of the administration to proceed and treat this like a new epidemic. And there are slides that I've had from long ago that show, if you draw a map of the U.S. and show a few dots in 1981 and see how that blossomed over the 8 years of the Reagan administration, it's incredible. Explosion of cases. And what's going on in society, as we talked about, there was a lot of fear among gay groups, and they were, I would say, mistreated. I mean, people just don't want, gay people going to certain public places in fear of catching something. And discrimination was a stigma, and discrimination were big parts of life for the gay population.
It's a counterfactual, of course. There's no way of knowing what could have happened. But is your hunch, if the Reagan administration did not approach this in cultural terms, but simply as a matter of public health and put the full resources of the federal government, would the AIDS crisis of the 1980s been much better contained otherwise?
Oh, there's no doubt we could have done a much better job, nationally speaking. And in fact, it was all the way until the Clinton era did the NIH funding take a real jump, and a concerted effort was launched. I mean, I think I still would give credit to the NIH leadership for directing a lot of resources to this, but it was obviously diverting funds from other areas into this new field. But there was not a massive infusion of support from the Reagan administration into this area.
Was Anthony Fauci at the center of all of this from the beginning?
He was already, I don't recall exactly when he became the director of the NIAID, but he certainly was in the middle of all of it. Certainly as the gay activists became organized, he was actually a major target for their critique.
Because he did not do enough, in their view.
Well, I think they always have to pick somebody. I mean, obviously, as a scientist, I know he was trying his very best with limitations. But they had to pick a number of targets. Of course, they protested Reagan and Bush, the first Bush, and the NIH director at the time, and of course Tony Fauci, because much of the funding to support this area would go through Tony's institute. So he was a target, but he initially kept the distance, but I think he learned over time that he could actually engage and participate in the conversation. And actually that turned out to be a very nice model for activism, engaging the scientific community to move agenda first. I think that's been the model, and the breast cancer community and many other disease-related communities have adopted that model, and actually together help push the FDA, for example, if there were a promising drug, to move them along much faster. Many of those would develop, the new paradigms would develop under HIV/AIDS.
All right, so now let's go back to the moment of discovery and the plan forward. So for you, what are those low-hanging fruit? What's there to work on next?
I think it's really, like I said, I was and is the physician scientists. So, the research lab do their own thing and the clinician do their own thing, but I could bridge the two. And that's why I could, for example, think about, what's going on in the clinic and bring it back to show that the virus is invading the testes, is invading the central nervous system, and a lot of clinical conditions could be explained by the virology, right? And then I could go on to show that within the brain, there are these cells that are called microglia, and they're derived from macrophages, and go back to the lab and show that macrophages are infectable. These are all things that we did for the first time and published before others did. In terms of other things, for example, there are lots of things that were already out there as agents to treat other viral diseases. So it was logical to see if we could take some of those and apply it to the new virus. And I could remember one of the early papers that I published that got a lot of attention is whether interferons were being used to treat hepatitis and they're general antivirals. So we showed that interferons actually could suppress HIV replication. So these are things that are almost obvious. You could sit down from day one to write a list of things to do. And these are the most obvious and most easily done. And so as a young researcher who now has a year or more of experience in the lab doing this, it's a no-brainer to do and to get the results out to the world. It's almost a bit like what we're going through with COVID now, because it's so new that there are lots of low-hanging fruits for the scientists to pick.
Because of your ability to traverse back and forth between a clinical and a research environment, what advantages did that give you? In what ways were you able to advance the research in general, having your foot in both worlds?
I think being a physician would cause you to ask the questions differently, right? You would ask certain questions from the perspective of a doctor, and that would be quite different from a person who's focused on the virus and then asking the questions coming almost from the other direction. I would just give you another set of example which occur temporally much later. Okay, so we also, and this I would say already take us to the 1990s. By then we recognized that there were some individuals who are infected but do very well. So the so-called outliers, 1% of them. Why? Now, you would only ask such question coming from the clinical side, because you are seeing these cases. Hey, doc, I've been infected most likely for 10, 15 years. My CD4 T cells are normal. I'm HIV positive by antibody testing, but I'm just fine. And we look at their blood, the virus is suppressed. They're not taking any medicine. And some of them are seeing at an era where we didn't have any good medicines. So what's going on with the outliers? By studying the outlier from the clinical side right now, right? You could get an appreciable understanding of what's going on. And it turns out that it's for them, and we study these people who are called long-term survivors, and get an understanding that in some cases, the immune system is uniquely capable of suppressing the virus. Now, not everyone could do that, but some of these could. And show that the immune system is actually making a pretty big contribution to keep the virus in check. Let me give you an even more dramatic clinical examples. Hey doc, I'm a gay man of say 45 years old. I have had 100 partners, many of whom have died of AIDS. And we had lots of unprotected sex, and yet I'm not infected. So this is another subset of outliers, right? And what's the reason that they're not infected? And it turns out to be genetic. So we studied these cases in the 1990s and show that for some of them, not all of them, for some of them, if you take their CD4 T cells from their blood and put it in the lab and try to infect with HIV doesn't infect. And up to that point, almost everyone's cells in the lab would be infectable. But these rare individuals their cells were not infectable. And it turns out that they lack the co-receptor molecule, a molecule called CCR5. So that discovery was made at our institute, simply from the clinical side. And by saying, well, wait a minute, why aren't these cells infectable? And what's different? And it turns out they are what we call homozygous defective for CCR5, the co-receptor. And that discovery was made as CCR5 was being discovered as the co-receptor via another approach. And we show that was absent. Now, interestingly, that also helps explain why some individuals do so well with infection. So these other individuals who are infected now, they're not protected, infected, but live quite a long time with HIV without treatment. And turns out they have one copy of the normal gene and one copy of the defective gene. And that's called the heterozygous state, right? So having one copy of the defective gene is protected. And so some of these questions not exclusively, but is largely coming from the clinical side. A basic scientist working in the lab would not be asking the same questions. And so we naturally would occupy a space, a sweet spot, so to speak, that would be good for doing investigation. And that's what my career over many decades has largely been focused. I don't necessarily have to compete with all the numerous strong bench scientists or all the people who are doing clinical trials and such things. I could define my own space and bridge the gap in many different ways.
David, with your interest in identifying why the outliers were doing well, more generally, were there advances in the field of genetics that were useful for this work?
In genetics, huge advances were being made, but up to that point, they were not being applied. Okay. Yes, I mean, it was mostly toward the Human Genome Project, right? Which came to fruition sometime later. And later, once the Human Genome Project was done, a lot of that technology was brought back to the HIV field to see why some individuals do well and some individuals don't do as well. And in the course of doing that, uncover a number of genetic factors, but the biggest one is still CCR5. And the other ones are immune molecules, right? Molecules that differ from person to person, but are involved in immune responses. But we already knew from the work done that some individuals have very robust immune responses and others don't. And so, yes, the modern genetic methods were brought to bear much later, but only confirming what we already knew.
David,
did not reveal anything new.
Obviously, at this juncture in the mid-1980s, you're operating in a fundamental research environment just trying to figure all of this out. What was your sense of the broader conversations about upstreaming, as we say in public health, prevention, public awareness, safe sex? What kinds of conversations were happening along these lines during this time frame?
A lot. I think, I don't want to take any credit for that. That was largely being done by CDC, NIH, and other people, and the media. And I would say I contributed my voice to that. I think the practices within the gay community changed so much in the first few years of the HIV outbreak. Having numerous partner was modified substantially. Having unprotected sex was drastically altered. The bathhouses were closed. So there were a lot of changes going on in society, and the message, and also the calming messages that, hey, this is not a respiratory virus. It's not casually transmitted. In fact, we also show, we're the first to show that there's actually very little virus in saliva. And whatever virus is there is actually not infectious. So there's something in the salivary secretions that inactivated HIV, even though the particle may be there, it's not infectious. So we contributed what we can to calm people about the lack of casual transmission, so that all this, a lot of the discriminatory practices were off target. If you're not having, contact with blood or sexual secretions, you're not at risk. So, whenever appropriate, we would help to lend a voice. But, much of the public health messages were being done by other people.
And at this stage, were pharmaceutical companies starting to think about drugs and therapies?
Yes. By about 1984, once the retrovirus was shown to be the cause, the pharmaceutical companies that had been working on antivirals jumped right in, right? And one of them, a leading antiviral company at that time, was Burroughs Wellcome, which has gone through many different partnerships and ventures. It's now part of what is called GlaxoSmithKline. But Burroughs Wellcome had a lot of stuff on their shelves that they brought into testing, and that's when. So they began that effort in earnest in 1984, and by 84, 85, certainly by 86, they found a number of compounds that worked in the laboratory and started trials on the first compound called AZT. And AZT was shown to be beneficial in patients for a period of about 3 months, and prolonged life by 3 months, which was substantial at that time, was a very significant finding. And then why it stopped working was not known until a little bit later, and that was the development of drug-resistant virus.
Now, was the approach of the so-called cocktail therapy, was that embraced from the beginning as the most promising avenue of therapy?
No, not at all. So AZT was the first to go into people, and as I mentioned, was found to be transiently helpful. Other drugs came along. They go by various names: DDI, DDC, interferon was tested, and so they were all targeting the step of reverse transcription. And they were all taken off the shelf from the cancer effort and from other antiviral effort. But they helped modestly from about 1987 to 1992. We would call that the dark period of AIDS therapy, where by then the caseload was heavy and the death rate was huge, and there was very little that impacted on survival. Yes, a few months here and there, but many clinical trials show that the benefit was minimal.
David, what about vaccines? Were people thinking about vaccines from the beginning? And was there a sense, even at the very beginning, that this particular syndrome, HIV, would be so difficult to target with a vaccine?
Well, there was initially euphoria when the cause was found, particularly when the NIH announced their papers at a press conference.
This is Gallo's work you're referring to.
Yes. The NIH director, Margaret Heckler, was there. And she, in fact, told the world, this will lead to a vaccine in a few months. And here we are 40 years later, we don't have a vaccine.
Did you have misgivings at the time? Did you share that optimism?
I didn't know what to think. I mean, I was excited about the discovery, and I knew I had a lot of work to do. But vaccine was probably going to be made by a company. And so that's not something we would necessarily focus on at that point. Yeah, many people thought that might be a bit optimistic, but hey, all of us were a bit optimistic at that time. But you know, in the historical records of the AIDS pandemic, her press conference proclamation is gonna go down in history.
Yeah.
Because that's been shown many, many times in the ensuing decades as how wrong we were.
Yeah, that's right. Scientists are fallible too, just like everybody else.
Mm-hmm.
David, last question for today. When was it time to move on from Mass General, and what were you looking for in your next opportunity?
Well, so I was at Mass General from 1982 through 1987. So, and during that period, you could say it was highly productive for me. I was walking around picking all the low-hanging fruits in the field. And by then, even though I had just started the postdoc, 3 years into it, I was sort of a new hotshot working on retroviruses at Mass General and Harvard. There were very few people working on that. And so essentially, UCLA Medical School and Cedars-Sinai Medical Center brought me back to be an assistant professor. They knew that I was, trained there, and my family was in LA, and so they made me an offer. And by then, Marty Hirsch has been a great mentor to me, and everything I did, I owe to him, because it was his lab, his resources, his wisdom that helped me, that allowed me to do what I did. But by then, I was the retrovirus expert within that institution, and probably at many other institutions. And so I felt it was time that I could run my own group and pursue my own research using my own support. And so the opportunity in 87 allowed me to do that.
There's a pattern in your career going back and forth from Los Angeles to Boston at this point.
Right. And then from there, I came to New York, and people had expected me to bounce back to California. But now I've been in New York since 1989.
That's right. That's right. Well, David, we'll pick up on that on our next talk. Thank you so much.